Rounding out our presentations today is Nine Diagnostics with AI-powered nanosensor technology for precision medicine and monitoring. Welcoming CEO and co-founder Freddy Nguyen.
I just want to thank the ILP and the Startup Exchange for having us today. So my name is Freddy Nguyen. I'm a physician, scientist, engineer by training, but I'm the CEO and co-founder of Nine Diagnostics, where we're really trying to work on determining treatment effectiveness, discovering new biology, using our AI-enabled nanosensor technology. Cancer diagnostics and biotools are evolving towards really more comprehensive profiling across the R&D and manufacturing pipeline, as you've heard throughout all the talks today. But some of the challenges are there's still limited biomarker insights, which really slows that real-time feedback that we can use. There's been poor stratification that's been driving the drug development failures, and there's fragmented data that really undermines the promise of precision medicine. And current diagnostics and tools that are being used are slow, expensive, and hard to scale. And so up until now, we've largely been limited to validated biomarkers. But at Nine Diagnostics, now we can unlock insights from unknown biomarkers and contextual clinical patient data. And we combine all of that together to create a new signature fingerprint using the technology at Nine Diagnostics. How does our technology work? It's based on single-wall carbon nanotubes, which you heard about earlier from Angela Belcher. But what we now can do to it is surface chemistry that allows us to introduce quantum well defects to those carbon nanotubes and really get a variety of types of carbon nanotubes. And then we wrap DNA sequences around it to provide that binding sensitivity and specificity around of the sensor. And we can detect everything from large proteins down to small molecules. But now you can see with all these axes of variables, we can have an unlimited library of nanosensors. And then we mix our sensors with a patient sample. Right now we use plasma proteins is what we're looking at. So looking at the plasma layer, we don't need any extra sample prep. And then we look at it through a high throughput near-infrared fluorescence system. And once we look at that interaction, we really can create these signature fingerprints. And then we start incorporating the clinical data from the patient, including their medication history, their coexisting diseases, their demographics information, social environmental factors, and then really develop that AI ML model in disease state classification. Some of our earlier data has been on developing screening diagnostics. One has been for high-grade serous ovarian carcinoma. The second has been in differentiation in the brain cancer space. And you can see, even with these two early validation studies, we've been able to develop assays with sensitivities in the mid 80 to upper 80%, as well as specificities in that 95, 98%. And all only needing about 200, in the first one, 269 samples, the second one, only 690 samples to use. And really a relatively short development timeline in terms of only 3 to 6 months. And so some of the more interesting and newest data that we have is that we've been trying to figure out, well, we can detect known biomarkers, but also unknown ones. So how do we determine what those unknown ones? So we've been developing the pipeline for determining what those proteins are. So from our most recent early brain cancer work, right now, we've been able to identify 37 proteins that our sensors responded to. And these are known to be associated with the tumor cells themselves, the tumor microenvironment, as well as the systemic immune inflammatory response. So that really allows us to do simultaneous detection and identification of relevant unique biomarkers without knowing what those proteins are ahead of time. And so that's really going to allow us to lead to new discovery and new biological insights and better understanding of what treatment response works. In terms of our technology, we use small volume samples. Right now, we can use just 300 microliters of the plasma layer, but we're working on trying to get that smaller. We can create these unique signature fingerprint profiles. We have a rapid assay development time of only 3 to 6 months. And it's a very cost-effective and rapid testing technology. Currently, less than $5 per sample in a research environment, less than 5 minutes to run per sample, and can continue to develop reliably high sensor performance. And so the way that we work with our partners is through a co-discovery process to build process analytical technologies or in vitro diagnostic tools, where we bring the AI-enabled nanosensor technology, our partners bring the biosamples, the contextual data. Together, we build these high-performing assays that can provide that actionable insights that you're looking for. Here's an example of how we've been working with one of our other academic partner at UCSF in terms of trying to think about endotyping to treat. So now really moving the paradigm towards how do we classify patients based on what they're most likely to respond to, as opposed to purely on the screening diagnostic side. And Nine Diagnostic aims to transform the R&D value chain to achieve that acceleration process, reduce the risk, and improve success rates. Right now, we're focusing on the discovery, the translational science, and the biomanufacturing part of the pipeline. So at Nine Diagnostics, we're trying to make smarter decisions across the R&D and manufacturing pipeline. Uh, by developing a high throughput proteomics and metabolomics platform technology, really for rapid biomarker discovery and response readouts. Our team has had 20 years of experience in this space. Over 20 plus patents have been filed in the nanomaterials nanotechnology space. And this is now the fourth startup that's spun out of my co-founder, Dan Heller, who is also an alum from MIT as well. So thank you so much for your time.